Hello there, my name is Luke McKeon, and for my Digestive Week presentation, I chose to do the anatomy and enzymology of fat absorption. So to begin, I kind of wanted to give an overview of the gross anatomy of the GI tract and all the sort of events leading up to the essential moment when the chyme or the ultimate item of food that you ate will enter the small intestine, where the majority of the fat digestion and absorption takes place. So to begin, you eat some food, and through mastication and contact with saliva, this food now has become a bolus and begins to move down the esophagus, as you can see on the right. And as far as fat digestion goes, this is actually happening to somewhat of an extent in the oral cavity through an enzyme known as lingual lipase. And after this bolus reaches the stomach and becomes chyme, it also will be being digested, the fats in it will be being digested there as well by another enzyme called gastric lipase. But because of some solubility issues that I'll talk about on the next slide, not much of that is really happening, and we'll have to wait till this chyme enters the small intestine for it to truly begin its fat digestion process. So as you may know, the chyme will enter the beginning of the small intestine known as the duodenum through the pyloric sphincter. And essentially, once it reaches this area, the liver will begin secreting bile salts, and the pancreas will secrete pancreatic lipase into this scenario. And these two factors will make for much enhanced fat digestion, and the enterocytes of the small intestine will take care of the absorption of said fat. So the essential role that makes bile salts effective in the aid of digestion of these fats is their amphipathic chemical characteristic. And essentially what this means is they have a polar and nonpolar region to their molecule. As you can see in the model I've included here on the right, detergent actually is essentially the exact same thing, and it's sort of going to take the nonpolar region and embed that within the grease in this case, which would be analogous to the fat in your body. And it's going to embed that, and then the polar heads of that are able to sort of mingle still yet with the ionic polar environment that would be analogous to the aqueous environment of your body. And so essentially this is only going to serve ultimately to break apart these larger fat globules into smaller discrete pockets, and these discrete fat entities will be easier for the lipase enzymes to act on now that they have much increased surface area. And now with this increase in surface area, the true enzymatic action of the lipase enzymes can take place. So in the book, triglycerides are noted as being 90% of the dietary component of fats that we eat, and I guess that's just because they are the most abundant and naturally occurring sort of fats that are within our diets. But nevertheless, lipase is a enzyme that turns these triglycerides into two free fatty acids and a monoglyceride, which for the enterocytes of the small intestine are much more manageable components to move into those cell bodies. And so it's essentially a hydrolysis reaction, and the main one that's going to be exhibiting action in the small intestine is pancreatic lipase. And there's actually a coenzyme called colipase, which, as you can see down in the bottom right in the yellow modeling, there's sort of some like steric hindrance around these smaller fat entities, And colipase works to essentially wedge itself in between those bile acid surrounding bodies to allow the lipase to more freely attack the fat, if you will, and break it down. So, and without this, it would be hard for the lipase to get in there and do its job. So, once these triglycerides have been broken down into their components, they're actually freely able to pass the apical membrane of the enterocytes in the small intestine. The model I have shown here on the right is a little strange. I pulled it from a paper that actually was more focused on the CLD body, which is cytoplasmic lipid droplets, which is sort of like a middle home base for metabolic processes concerning fats inside of cell biology. But in any case, this still serves as a very good model for the more anatomically concerned process of fat absorption. So essentially, again, the components of the triglyceride that have been broken down by the lipase enzyme in the lumen, which would be on the left of the model, can freely pass that apical membrane, as they are also nonpolar, just like the plasma membrane itself. And once they have done this, they're going to work their way into the smooth endoplasmic reticulum, where they will essentially reform triglycerides all over again. And then they will also combine forces with cholesterols in that endoplasmic reticulum, as well as proteins. And these three heavyweights will essentially combine into a larger structure called a chylomicron. And these chylomicrons are in fact so large that they need to be sent over to the Golgi to be packaged for exocytosis. And also, because of their large mass, they're not going to be entering into those capillaries inside the villi of the larger or the small intestine anatomy. They're rather going to go into lymph lacteals, which will ultimately carry them back into the superior vena cava, and that's where they're going to re-enter the systemic circulation. However, it is actually possible for smaller free fatty acids who have not joined up into these chylomicrons to freely pass the basal membrane of these enterocytes and just enter back into the bloodstream because of their small size, and they do not have the need to be exocytized. But here are some of the two sources I used to construct this presentation. But in any case, I thank you for watching.